Welcome to the Providence College Podcast. Subscribe to the show on iTunes, SoundCloud, Stitcher, and Google Play. If you like what you hear, please review and share with others. Email podcast@providence.edu with questions or comments. Go Friars!
Hello and welcome to the Providence College Podcast. I'm your host, Matt Chittum, and I'm joined, as always, by PC producer and videographer Chris Judge, class of '05. Here at the Providence College Podcast, it's our job to bring you interesting stories from people inside the Fryer family. And today we are bringing you Dr. Victoria Templer, Assistant Professor of Psychology and Principal Investigator in the Neuroscience and Animal Cognition Lab. We'll be speaking with Dr. Templer right now. In the second part of the podcast, we will be speaking to Senior Keith Lee, who is a biology and psychology double major and also works in the neuroscience department. So, first of all, thank you so much for joining us, Dr. Templer.
Thank you for having me.
It's my pleasure. And I guess the first thing is, what is the Neuroscience and Animal Cognition Lab?
So that is my lab that I started here just a few years ago when I started at PC. It is called the Neuroscience and Animal Cognition Lab for obvious reasons, but what that means is we are interested in the biological and neural bases of behavior. So we're interested in neuroscience, right? So we're interested in studying the brain, right? And we're also interested in animal cognition. So animal cognition is a broad field, but what the questions that we're asking are about comparative cognition, which means we're interested in studying non-human animals and comparing them sometimes to other non-human animals and also to humans. So that's what we mean by comparative, and it's cognition because we're studying the thoughts and behavior of non-human animals.
Got it. So how do you, this is probably too broad of a question. So how do you study the cognition within an animal versus in a human?
That's a great question. So what we like to do is develop tasks, which it can be kind of tricky to develop sort of clever tasks that allow us to look at objective, observable behavior in animals. So we can't ask an animal, what are you thinking, right? We can't just animals can't verbally respond like we can, but the same thing you could say is true for a 1-year-old or your young child, right? So they don't have any verbal ability quite yet, or they might have limited verbal ability, but it doesn't mean they don't have thoughts. And so the question is, can we look at their behavior, their particular responses to different tasks and use that behavior to basically differentiate, is it this type of thought process that's going on or this type of thought process? What mechanisms, what neural and cognitive mechanisms are explaining their behavior and their sort of mental representations? Are
there certain levels within this animal community where you feel like their brains have developed well enough where you can actually make a comparison to a human? Like, where's the line?
So that's a great question, and that is the reason that neuroscience and animal cognition, in my opinion, are so intricately linked, because actually the brains of especially non-human primates are very, very similar to ours. So that's the field that I was coming from. I used to work with monkeys, and I made the transition to rats when I came to PC because, frankly, it's a lot easier to do neuroscience on rats, and I didn't want to open my own monkey lab. But the brains of rats are also actually very, very similar. So there's a lot of functional and structural homology. And so what we can do is actually look at the brain structures in rats and humans. Say we take the medial temporal lobe. This is a part of the brain that is involved in memory. And a lot of the substructures within that are really, really similar in humans and rats. So the same types of neurons are in those areas. And so the neurons that become activated and the particular brain regions that become activated, say when a rat encodes a particular environment, right? And then maybe later it remembers that environment, the context it was in, when that happened, what happened, are actually activating the exact same brain regions that they do in our brain. So that is one reason that I think, studying non-humans is so important, both for the human translational question, and also just because understanding how non-human animals think, what's going on in their brain is interesting inherently, just for a basic science perspective.
So is the memory portion called the mid-temporal?
Medial temporal lobe,
right.
just
medial
one
temporal lobe.
Part of the brain, yeah.
All right, you'll have to bear with me on some of this stuff. So that aspect of the brain, is that again, pardon my ignorance on this subject. So is that the part of the brain that's a little bit more on the developed side, or is that kind of like the lizard brain part where, it's kind of a basis throughout the animal kingdom?
Yeah, so great question. So, that's just one example, the medial temporal lobe, and the hippocampus you maybe heard of is inside the medial temporal lobe. That's one region that we know is, very similar, but a lot of regions in the brain are. And what people think of as kind of the reptilian brain, like your brain stem, we share that with most mammals. And, the brain sort of evolved from the inside out. So the higher you go in the cortex, we have more association areas, sort of higher out in our brains, or more towards the surface or dorsal than other species. But a lot of those structures are the same, that are what you might think of as the higher cognitive areas, not just the basic, what you might call reptilian brain or older brain that's been there for a long time in our common ancestors and other species.
Right. So we're talking about rats right now. You just brought up metacognition and things like that. So you recently, with the help of some of the students here at PC, published a paper that got a little notoriety around here, and I found it to be just a fascinating subject. Can you mind just, what exactly was the basis of the study, and then we'll dive into it?
Yeah, so we were interested in asking if rats can be aware of their own memory states. So we call that metamemory. You could also use a sort of more broad term, which is metacognition. And all metacognition is really just thinking about thinking. So it's interesting because this is what we think has enabled humans to have things like consciousness, right? So you can have a really, really rich definition of thinking about thinking. It comes with feelings of having been there, and we have all these personal anecdotes to add to it, and consciousness. But at its most basic level, metacognition is thinking about thinking. So you can ask if a human or anyone can be aware of their own memory states. So can you detect an absence or a presence of memory? And so that's sort of making an operational definition out of this hypothetical construct of metacognition, right? That could include consciousness, all these more sort of interesting things, but it also could just be at its most rudimentary form. Can you be aware of your own mental states? And in particular, I'm interested in can animals be aware of their own memory states? Can they identify when they've remembered something and identify when they've forgotten something?
And then within that, it's not just one or the other, right? There's kind of the range of how confident are you, I guess, in your memories? Or like, all right, a scale of 1 to 10, I know that happened because I just did it, versus I think I might have done it, or I think I might have thought about that at one point. Yeah,
so so human researchers are asking these questions because it gets very intricate, as I think you're leading to. It's not just, I remember, I forget. There's a huge continuum of familiarity cues that you get. You get familiarity cues. There's a difference between, there's a whole field of study where people are asking the difference between knowing and remember. You just know something, or and there's different confidence levels. So, people have reported, you've had monkeys reporting their confidence intervals. They'll bet how much sort of tokens they want to wager on if they're really sure they were correct, or they're not so sure. It's kind of like Who Wants to Be a Millionaire. There's a lot of scenarios that are developed almost off of that paradigm in animals. So my question, which, had only been asked by a couple researchers, and was that do rats have the ability to be aware of their own memory states? This question has been asked in other species. It's been asked and I worked on this while I was in graduate school, but these questions have been asked in a number of different ways with different measures. In monkeys is probably the widely studied species that besides humans. And so the rats was a really, really underdeveloped area. So I thought, why not just come in my first study here and use rats, which was my new model organism, and ask this question.
So how do you go about, setting up the experiment, and what exactly did you do for the rats or with the rats to get to that point?
Yeah. So let me, I guess I'll explain. I'll back up and sort of explain what I like to give as sort of a human example, so you can understand how the paradigm in rats works, because it is a little complicated, and I can't draw pictures here or anything, or use a whiteboard. So in some tests in humans, I think the SATs used to be like this, maybe in our generation, but not in students' generation, where you were allowed to leave a question blank, And if you left a question blank, you wouldn't get plus or minus any points. But if you answered it and you guessed wrong, you have, say, 4 choices. So you have a 25% chance of answering correctly. You would get, if you answered incorrectly, you would get minus a quarter of a point. If you answer correctly, you get a whole point. So obviously you want a whole point all the time. But there's times when you know you don't know the correct answer. So in that case, it's pretty wise to leave the question blank. And you don't get penalized for that, right? So what we did is we gave rats a similar situation where they're allowed to opt out of a certain memory test. We give them a memory test and they're allowed to opt out of it and say, you know what, I don't want to choose A, B, C, or D, right? I just want to choose a less preferred but guaranteed, what we call a less preferred but guaranteed reward, which is sort of equivalent to that plus or minus 0 points. So what we had the rats do is we had them sample an odor. We needed to give them something to remember. So they dig in a little cup. Keith and a few of my other students helped train rats to do this. So they dig in a cup and they smell a particular odor. So say it's cinnamon. They smell cinnamon, they dig in the little cup, and they get a Fruit Loop at the bottom of that cup. So they know, they're trained that if you keep digging in a cup and you're right, you'll get a Fruit Loop. So they get a Fruit Loop during sample, just so they're reinforced to keep going on to the next phase of the trial. And then they reach a decision point. They enter a room, they sort of wait in this waiting chamber after a delay, so they smell a sample, they wait a particular delay, and then they enter a room and they can either turn left. And turning left means I want to take the test. So I want to choose which of these 4 cups. There's 4 cups, and they all contain sand, and they all have different scents associated with them. One of them, cinnamon. That's correct. So if you get it correct, you get a full Fruit Loop. But if you get it incorrect and you choose thyme or you choose paprika, whatever the other scent is, you're going to get nothing, right? And the trial's over, and you just get to go to the next trial. But at that decision point where they can turn left and choose one of those, dig in only one of those 4 choice cups, they're also given the option to decline the test. So in that case, you'd turn right and you get to dig in a cup filled with sand. It has no odor. It's actually a different colored sand and a different floor texture. So the rats really know this is a different situation, right? This is a different answer. And that provides the rat with I think it's a quarter of a Fruit Loop. So it is less preferred, but it's guaranteed. It's always there, right? That quarter of a Fruit Loop is better than nothing. So that's sort of the contingencies, if that makes sense. The rat gets to sample something, and sometimes it happens to remember, sometimes it happens to forget. We titrate the delays in between the study phase where they get to sample that sample odor and the test phase where they get to take the test, such that performance is in sort of a sweet spot of about, 60-70%, so that they're not 100% right, because then you're always going to want to take the test, right? And then it shouldn't be at floor, it shouldn't be at ceiling, it shouldn't be at floor. At floor, you should probably always decline the test. So we make memory a certain strength so that sometimes you just happen to forget, sometimes you happen to remember. And so the rat smells the sample, they have their delay, their individually titrated delay, and then they get to choose if they'd like to take the test or if they'd like to decline the test and get a less preferred but guaranteed reward.
So was there any variance with the rats in terms of the wait time that affected them? Or was it just generally like, all right, if we wait this long, that will work on all the rats, or some rats had maybe different memory strength?
That's a great question. And yes, there is a lot of variability. I'd say the average delay time was about 1.5 minutes for the rats. So this project, we're very proud of the work that we've done, but a lot of times there's a whole backstory to the fact that you'd actually did 10 times more work than is actually published in the manuscript. That's common for scientists. So the 10 times more is actually, I really credit my students, especially Aidan Preston and Keith Lee, for doing this. But we had about a year before we started collecting this data included in the manuscript where we were titrating delays, and the rats' memories were just too good. They were remembering for over 20 minutes, which meant my students, to test 9 rats, had to be in the lab all day and not even get through all the rats because there's multiple trials in a session. So their memories were just too good. They could remember for a really long time, right? And remember that we're giving rats their most salient sense, right? So we're giving them odors, that they're really, really good at detecting odors. If you were a human, if you were a monkey, I'd give you a visual task. I'd give you a picture of something, match the sample picture, a picture of a star. You'd have a delay after your study phase of seeing the star, and then at test you'd have a star, a circle, a square, and a squiggly line, right? And you get to match to the sample, right? But for rats, we're going to give them odors. So this is odors are particularly salient for rats. So I think that contributed to why their memories were so good. So what happened is we needed to use little tricks of memory based on what we know about memory mechanisms to decrease their memory, to get their delay to be about 1.5 minutes versus 20 minutes.
Like what? Like what would you do?
So what we did is originally we had what we call a set size of any of the odors that could show up on any of the trials was like 20 odors, right? So you could get, cinnamon on the first trial, and then you could get, something else, but the whole set size that you draw from on each trial is 20 odors, right? So there's, I guess so. So what we did, so one thing that we did is we decreased the set size so that it was only 4. It was only 4 odors. That means that on every trial, you are smelling the same odors. You'll have a different one for the sample, perhaps, but each test phase, when you get to choose which one you sampled a few minutes ago, you're always going to have the same 4 odors, say lemon, paprika, thyme, and cinnamon. You're always going to have those exact same 4. So what happens in that case is something called a high memory interference, which means that I'm speaking anthropomorphically, but if you're a rat, you're not going to know if what you just smelled happened on this trial or the trial before. So you're smelling lemon and you are presented with all the 4 odors and you're like, lemon smells kind of familiar. I must have just smelled that. Turns out you might have smelled that 2 trials ago, not the present trial. That's called interference, right? Because you don't, so you're more likely to make errors. So it decreases the performance because there's more interference between episodes.
Right.
Does that make sense?
Absolutely. Then that way you're able to gauge, the level within, their own head, the level within their memory of how much they can pick apart what they've experienced.
Exactly. So it gets fuzzy. Basically what we're trying to do is make that memory representation a fuzzy cue. So the other way we can make it a fuzzy cue is we can decrease the amount of time in between trials, right? So if you give you've trial 1 and then 10 minutes later you have trial 2, it's gonna be more easy to distinguish that, okay, I just smelled thyme at this sample on this trial, right? It seems like it was a long time ago that you gave me lemon 10 minutes ago, right? But if you put the trials really, really close together, right one trial right after the other, that also is going to increase interference. Right?
This is so interesting. So this really gets into the idea of, you really have to spend a lot of time planning these out, and like the amount of problem solving needed here.
Yeah.
Between everyone working on this project, it's like astounding how much time you have to spend really knocking out all of these details. Yes.
It's a lot of effort, but it's fun too, and I think the students learned a lot, so that's what's important.
I can imagine. So if I was doing a memory test, now I'm not a rat, despite what Chris says over here as my producer. No, if I was doing a memory test, right, you said before, I'm a human, you'd give me the visual, I have the star, then match that up with the sun, the squiggly line, and so on and so forth. If I messed it up, I would probably have some sort of it would be very obvious that I was disappointed or I had regret, things like that. Did you ever see any of that in the rats? Or are you able to read that sort of thing?
So that's a good question. Keith would be the better one to ask it because it was my students in the front lines actually collecting the data. But I think they did, they did notice that. So for instance, the rats usually once they made a decision to turn left or turn right, I want to take the test, take the memory test, or decline it, they would stay there. But once or twice they did notice a rat sort of start to smell all of them. And then it was almost like they decided, again, I'm speaking anthropomorphically, which means I'm giving, human attributes to animals. But, it almost seemed like they they said, I don't know, I don't recognize any of these from the sample. I don't know which it was. So then they traveled over to the decline option and sort of bailed out of the test after, which is interesting in itself. But you do ask a very, very good question. We didn't test for that. I know that we actually call them behavioral cues that animals do engage in. So, for example, monkeys we know vacillate, and I would see that. Monkeys would get when I was testing monkeys in graduate school, they'd get very upset sometimes, or they'd vacillate when they don't know and sort of get like anxious or frustrated. But that, interestingly, can actually be an objectively observable behavioral cue that could be cueing them to take the test sometimes and decline the test other times. So what we try to do in these experiments, what we tried to show is that what the animals are doing is actually based on an internal cue. And the internal cue is introspection. That's, judging the own strength of your memory, right? But what we try to do, and why we did so many experiments in the study, is we try to rule out the possibility that animals were, instead of responding to that internal cue, they might have been responding to an external cue in their environment, even like their own body moving or starting to get anxious or something like that. Sort of similar to, a response latency. So if you're taking a multiple choice test, there's a certain amount of time that you might wait before you start circling in A, B, C, or D, right? So if you have a really long response latency, that's generally associated with less correct answers.
Right.
So even though you have metacognition, I'll admit you have metacognition, I know you can introspect, you might be using other cues even if you're not aware of it. Like you might not even ask yourself, I know I don't remember the Pythagorean theorem, so I'm going to leave this one blank. You might not even need to say that.
I do remember the Pythagorean theorem, Dr. Templar.
That was okay. Well, I don't remember how to do it. So what I would do is I could say to myself, okay, I know I don't remember this, and I could leave it blank, but I could also choose an easier way out, which doesn't necessarily mean I can't introspect, but I could just say, without even being too aware of it, just knowing that I took a long time to respond and just leaving it blank. So I'm actually responding to my response latency as the behavioral cue that is guiding my use of that decline response or that ability to opt out of certain tests.
That makes a lot of sense, and it actually has a direct comparison with being a trial lawyer. Not that I ever was one, but they say that if you're ever on trial, that you should always answer the questions in the same cadence. So not say for me, they say, what's your name? And I just immediately fire back, Matthew Chittum.
Mm-hmm.
What's your date of birth? January 24th, 1981. Because if they then ask a more detailed or pertinent question, and all of a sudden I'm taking 3 or 4 beats between the question and my answer, like, where were you on July 4th? And pause.
Right.
I was,
Right.
It kind of gives it away that you're obviously not very confident in what you're saying. Exactly. So that does make a lot of sense.
Yeah, exactly.
So when you basically concluded that there was metacognition or metamemory within the rats, were you able to say like what level or how much metamemory, and then also what was the overall goal after you determined that in terms of the transferability of that study?
Yeah, so great question. So I think I should back up first because I don't know if I specifically went over it. Explaining the task that we use and the contingencies of, you can take the test and you can sort of see if you can get it correct and get a full Fruit Loop, but you only have a 25% chance of getting it correct if you're just guessing, right? But hopefully you know the answer and you're going to choose to take the test when you know the answer. So what we do is we compare something called choice trials. Those are trials when they have the option to decline it. They have that cup that's like the safe option. You always have a small piece of a Fruit Loop in that cup. We compared those choice trials where they have the option to decline to trials on which they do not have the option to decline. So those we called forced trials. Those are just basic memory tests. That's like taking a test where you have to answer every question. It would be silly to leave a question blank. You should at least guess. So if I were to give you a test where you had to answer each one and I don't give you the option to leave them blank, your performance is, if you have metacognition, if you can be aware of your own memory states, your performance should be better on those tests where you have the option to opt out, right? Because if you have metacognition, you're going to opt out of trials on which you were likely to answer incorrectly. if I don't give you that option, you have to answer every one, that's going to bring your score down, right? So what we do is we compare chosen trials and forced trials to each other. And if you are significantly better on those chosen trials where you have the option to actually decline than you are on sort of your basic memory test, your forced trials where you just have to choose one of the 4 answers, that is sort of indicative of metacognition. That is our observable behavior, that is our operational definition of metacognition in terms of being able to detect the absence or presence of memory.
That is genius. I was before I even read this paper, I'm thinking like, all right, how is this even possible? And then just hearing you explain it like that, it just makes all the sense in the world. It seems, I guess that's the whole point. It seems obvious after the fact.
Yeah. And I should be clear that I didn't develop this paradigm. I used the similar paradigm in monkeys, but other researchers, including actually my graduate advisor, helped develop this paradigm to test observable behavior. Because your first question, I think one of your first questions is, how do you ever know the mental states of animals? And it's tricky to develop paradigms that can really get at complex forms of cognition. So I also study things that perhaps aren't as complex, like mazes and spatial navigation and working memory and other things that you can use more standard, mazes that you would employ to rats and mice and people do all over the world. So we do things like that, but to get at really intricate aspects of cognition that really sort of tease apart what are the representational features of a particular mental state, it's difficult to develop sort of clever paradigms that do that. So hopefully that design helps explain how we do the type of work we do in the field of animal cognition.
No, absolutely. Now, is there, I guess, kind of staying on that topic, are there anything that another researcher could point out and say like, hey, there's another explanation for why there's a differentiation between the scores? Are there other potential variables? Great
question. So yeah, I think to sort of answer the question that I didn't completely answer from your other question is, so what do we do after we have that difference? And okay, rats are better. Yes, they are significantly better on those tests where they have the option to opt out or decline certain tests. It shows that they are in fact using that response adaptively or appropriately, which means they're declining trials on which they would have gotten incorrectly, right? So we have that. Okay, so we have our demonstration of metacognitive behavior, but we're not satisfied to say rats have metacognition. That's the be-all and end-all. It's a very, very complicated question, and it's not an all or nothing. We're really determining more acutely what is the extent to which they have this objective behavior and what are the other things that it could be. So what we like to do, or the way I generally test this, is to think of all the options that could be cueing their behavior to adaptively decline trials on which they were unlikely to answer correctly. Right, so that we just think of chosen trials are better than your forced trials because they have the option to decline. So there's a few different cues that it could be. So the most interesting one, of course, is introspection. That's using an internal memory state that sort of cues my behavior to say, okay, this is a good one to decline because I know I don't remember what I just sampled on this sample. I don't remember what scent I sampled when I was digging in the cup during the sample phase. So that's introspection, but we can only infer that if we rule out external sources of stimulus control. So that's internal, right? Introspection. There's external cues like we were just speaking about, like response latency or behavioral cues, things that over time you could learn to associate with a particular response. So you would get more rewards over time. Even if it occurs in the absence of any awareness, over time, you could simply learn by basic rules of conditioning and learning that anytime you take a long time to respond, it's best for you to opt out of that trial type. It's just an association. It doesn't necessarily require any internal monitoring. It's sort of something that anybody else could observe. Like you were saying in the courtroom, you could observe somebody having a high or a low or a long or a short response latency. Same with if you start to get nervous and you don't know, you'll start, or a monkey at least would start to, sway back and forth. So that's an external cue that anybody could know about, right? But your internal cue is private. Only you should know that.
So like if the Fruit Loops were always in the cup on the right-hand side of like if they turn left, they go into that chamber. The Fruit Loop's always on the right-hand side, irrespective of whatever the smell is.
Right.
Over time, they'd
Right.
be able to figure that out, and they can just, that would be the
Right.
the variable.
Right. So we're very careful to counterbalance things and make sure that, it's not always going to be in the same cup or the same location. We do a whole, complicated randomization procedure to make sure that they can't, because rats are just like humans. They're going to choose the easiest way out. They are across the board. They're going to find the easiest way. It doesn't necessarily mean they don't have the more interesting mental representation that we're really, more invested in answering, something like introspection, but they will take the easiest option out. So what we do is we see that, performance, we have the metacognitive behavior. They're better on those chosen trials where they have the option to decline than they are on trials when they don't have that option. And then what we do is we test for, like you were saying, generalization or transference of that adaptive use of that decline response to different contexts. So different contexts are really important because you can't, there are less cues in the environment that the animal could associate with particular responses over time. So, we did look for things like, were they more correct on, did they have biases to certain cups or certain odors? And that doesn't seem to be the case. What we did next is we increased memory and decreased memory. So when I was talking about the internal private cues and the external publicly available cues, if we manipulate the memory, which is only an internal cue, there should be nothing in the environment telling the rat that this is a long delay or a short delay. But what we can do is the delay that they usually have, we can make really long and we can make really short. And so if the delay is really short, they should want to decline those trials pretty infrequently. They should want to take the test all the time. And that's what we saw. We also saw the opposite. If it's a long delay, they should want to decline a lot at the time, right? And that's what we find. So what we do is we manipulate memory in multiple ways and see if they are likely to transfer that ability of adaptively declining those tests to other contexts, where there's no common sort of external stimulus features that they could have associated and cued them to choose the decline response in different contexts, if that makes sense.
No, absolutely, because you also have the situation where, like, say, if I'm a rat and I don't like paprika-flavored Fruit Loops, and all of a sudden I smell paprika, I'm like, no, I know the answer to this question, but I'm not going to answer the question because I don't like that.
Right.
Versus like, hey, cinnamon-flavored Fruit Loops, like, that's great.
And that's an easy rule. So we can so the question is, do they associate things? Do they just pick up on easy rules? And the reason that we tested them in so many different follow-up experiments is to see if they were following a rule like that. And the rats didn't seem to have a bias for particular odors, and they seemed to remember all of them similarly. So we looked for all those things, and also tested them in multiple manipulations of memory to make sure that their responses were associated and consistent with their memory state, which is an internal cue rather than an external cue that is not based on real meta memory or awareness of your own memory state. And
when you look at those findings, how do you extrapolate out to the comparisons to humans versus, like, all right, how can studies like this be valuable to human memory, and how does this study in particular kind of help that cause? Yeah, so good question. So I think in general, non-human animal studies are interesting because they actually help us better define our definitions of these constructs, even in humans. So in humans, sometimes I'll just assume you have consciousness, right? It's sort of like, the Turing test or a question of, do robots have this awareness, right? So I could assume that, and you can give me a verbal report. You can tell me, what it's like to be, you could tell me a memory you have and what it's like to be aware of that memory. But sometimes those verbal reports are fraught with biases and kind of unscientific, data that is not quite as objective or observable. And not to say that there's anything wrong with assuming that you have consciousness, but we don't just want to assume it is the point. We want to actually look at objective, observable behavior. You might imagine somebody that has a particular brain abnormality, and perhaps it's not possible for them to have consciousness.
And so we shouldn't just assume that everyone has a certain mental ability. We need to have better criteria and definitions for it that we can test across different species. So I think that's one piece of it. And I think the definitions of metacognition have even changed in humans because of these animal studies that I would say are very, very rigorous in their definitions of mental states. And there are a couple other reasons. That the other main two that this is interesting and important is because of its translation to humans. So like I said, the brain areas in rats are actually very, very similar to humans. So if we have these models of these types of memory that are really important and interesting, and they degrade with dementia and different forms, Alzheimer's, different forms of dementia, then we have a model that we can employ in future studies that are perhaps more translational, like drug discovery treatments in tapping into these special types of memory, like metacognition. So that's sort of an anthropocentric answer, which is human-centered, right? It just means that you're asking a question and looking for an answer so that we can better treat and understand the neural and cognitive mechanisms of memory and particular types of memory like metacognition in humans, so we can improve our treatments for it in the future and better target those particular types of mechanisms that become degraded with old age or with diseases like dementia. So that's really important, and that's one of my major motivations. And then the other reason is just, I sort of call this my National Science Foundation reason, which is just the other one I would say is more NIH, but the National Science Foundation is just that it's interesting in and of itself, because how rats think and what their mental states are they capable of metacognition, I think is a fascinating question in and of itself. And these questions and the answers that we are able to get from doing these studies do shed light on the evolution of intelligence and when these particular forms of cognition evolved. So were they really just evolved just for humans, or are there rudimentary forms of metacognition that were perhaps there and evolved for different reasons that might have served ancestors that we share with non-human primates and with other mammals like rats?
There you go. So what's next?
What's next? So I would love to continue researching metacognition, and I do have ideas for new studies that would frankly be pretty easy for us to carry out. But I do have a lot of other research programs going on right now, and they're not actually about metacognition. So I was fortunate enough to have a grant, so we're doing some work over at Brown, also because of construction. So that's on a different project about top-down and bottom-up processing in rats. We're also doing some aging studies. So we've got a lot of other things going on, but right now I don't actually have the follow-up study. I have the idea for it, but not necessarily the time or resources to do it right now, but maybe in a couple of years.
But the research continues, just in other areas.
The research definitely continues, yes. We are very busy in the NAC lab.
That's wonderful. Well, thank you so much for joining us and taking a deep dive into this. This is fascinating, and I'm excited to talk to Keith about this as well.
Great. Thank you for having me and for listening to all these details of rat memory that you might not have wanted to know.
No, I did some reading beforehand, so I would be able to, react in real time, but I do appreciate it. So thanks a lot.
Thank you.
Welcome back to the Providence College podcast. I am here with senior Keith Lee. Keith, thank you so much for joining us.
Thank you for having me. It's good to be here.
Well, we're fortunate to have you and Dr. Templar here today, and we just kind of got into all the work that students like yourself are doing here in the sciences, and it is really very interesting stuff. And before we get into that, just touch on some of your background a little bit. You're a bio and psych double major, as well as a neuroscience certificate, taker? How do you say? You're going to be completing that? Or
I will be completing the program, yeah.
Okay.
Hopefully, if all goes well.
And then you're from Doylestown, Pennsylvania, as you mentioned before. So you're going after a couple different things academically. How did you make the choice, what to major in, what to double major in, and all of those selections that you have to make either during the application process or once you're here on campus? When
I was applying to colleges, I kind of knew I wanted to go more of the science-y route, I guess. And I came in as a psych major. I took a psych class in high school and loved it. Figured this would be sort of a cool area to explore. And when I got here, that's when I learned about the neuroscience program, and I took human neuropsych, which is a class in the psych department. And that's when I started to explore more neuroscience and get more into the bio aspect. And that's been one of my favorite classes, the human neuropsych. So kind of from that is when my interest started to move into more neuroscience. So then I started taking more bio classes, picked up the bio major, and yeah, here we are today.
So how much did your high school psychology class, serve as a foreshadowing of stuff you'd look at later, or was it just kind of a general take?
Just a very general take, kind of exploring all different aspects of psychology. So it hit on sort of everything. But yeah, it was a nice, I guess, foreshadowing of what was to come.
So what is the interplay between biology and psychology for you in some of your coursework, or how you look at the field in the future?
I think, that's kind of a difficult question, but I think psychology, I sort of view psychology more in terms of biology, like how our thoughts come from biological mechanisms and how our behavior comes from biological mechanisms and how sort of that all works into environmental factors. So yeah, that stuff is certainly more interesting to me than psychology that deals more with maybe social psych and stuff like that. So yeah, I find that just more interesting and more connected to the biological aspect. And
was that always the case, or did you kind of come around to that thinking once you started taking some biology classes or got more ingrained in the psychology major?
I don't think it was always the case. I feel like in high school, they were two sort of separate fields, biology and psychology. And in my psych class in high school, we didn't do a lot of biology. It was certainly more social psych and things like that. But then when I took like human neuropsych, that's when we learned about the different areas of the brain and neurons and that sort of stuff. And that's when it sort of became more of a bridge together. And I fell in love with it. So.
Right. So for you, is it hard to not analyze your own thinking or the thinking of others as you learn more in this field?
Sometimes I feel like in class, that's when I'm like started to really think about how I operate and how I think, and how others think. But once you kind of leave the classroom, I'm like, don't think about it too much. Yeah, because you kind of go crazy, I think, if you do that too much.
Right. There's always that anecdote of people who go through med school who at some point during their studies have come to realize that they have every single disease.
Yeah.
So you just brought up a good point. So once you leave the classroom, so for, I guess, the evolution of your classes here, how they started in terms of the traditional classroom setup versus being in the lab from freshman year on to now, your first semester, senior year classes.
Freshman year, I took classes that had lab components, Gen Bio, for example, lab component, all that sort of thing. And then sophomore year, we have RDSA, which is a psych course, full year, and which there's also a lab component. And that's where you sort of learn how to do more psychological research, analyzing data. So that was really cool. And then sophomore year is also when I applied to start working in the NAC lab with Dr. Templar. And that's when it sort of all kind of changed and my perspective broadened a lot more. Got to do more hands-on research, learn a lot more literature review and things like that, and really learn and understand how to conduct research in a real setting. Because the lab classes, it's fun and it's good, but it's very different than what you actually do in conducting research.
And how quickly do you get up to speed in terms of the lab processes, in terms of being competent in the lab and then taking it a step or two steps further where you're really engaging in some high-level research like y'all did this past year?
I feel like it happened all pretty quickly. Like I started joining the lab and got to meet everybody, did some reading of what we do and what we're interested in, and then pretty much jumped right on the project and started helping with testing and data collection. And then it was seniors above me who taught me how to analyze data and do all those sorts of things. So it happened all pretty quickly, it seems like, and then it was seniors who helped me to learn what to do and really get my feet under me so I kind of knew what was going on.
So how does that differ, I guess, when you have a senior teaching you versus a professor? What's the different dynamics like, and what are the different experiences like?
I guess, I don't know. It's different in that I can relate to him more. Like we both have similar schedules. We're both students, so we both are sort of dealing with similar issues in terms of maneuvering college. So he's also kind of helping me think about what do I want to do post-grad life, because I'm seeing him do that. But yeah, also, it's more of a casual relationship, I guess. But yeah, it was, I don't know.
Right, and I know it's one of those things as well where I feel like you, especially from a student's perspective, once you're able to teach another person, it really must ingrain itself in you. And as a senior now, I wonder if you're going to be in a similar situation where you're going to help mentor other people. Yeah,
I mean, I would love to, hopefully so. Yeah, that would be really cool.
Got it.
Give back and keep the tradition going, I guess.
So you mentioned just now, so you're thinking about your post-college stuff. Is there anything that you're working on specifically, or what's your mindset for approaching that step?
I'm kind of all over the place, sort of. I'm certainly interested. So the summer before, I worked in the lab and did research, and I loved it. This past summer, I interned at a hospital and did more clinical stuff, looking at the medical field, and loved that as well. And I haven't really locked down on anything. I'm thinking maybe going to medical school. I'll probably take a gap year and do some work, or maybe do research for a year, and then maybe look at applying to med school or maybe grad school for psychology.
So what is the thinking for students generally, and then you specifically, in terms of the positive benefits of taking a gap year versus just hopping right into further education?
Well, I think it's especially important if you're not exactly sure what you want to do, because it takes a lot of time and a lot of money and effort to apply to schools. And if it's something that you don't want to, if you realize once you get there, this is not for me, that was a big waste of time and effort and money. So I don't think it's worth it to maybe waste all those energy and resources if it's not something you're totally locked into. And also maybe to get a feel for something other than what you've been doing, to work in a different setting, to, really sort of clarify what you want to do, and discern maybe what you want to do and for the rest of your life.
That's a great point. And I feel like when people get kind of the outside the college experience, whether there's an internship or a gap year or working in the summer or a whole host of things, it really not only can help crystallize something you might already think, but it can also, in some respect, help you cross things off the list, right?
Exactly.
That sometimes can work
definitely
out pretty well.
Yeah, which I guess it kind of disappointing sometimes when you go into something, you're like, oh, I hate this. But I guess it's just as important to be like, oh, well, this is something I'm not going to spend more time into or do for the rest of my life. So yeah, just as important, I guess.
That's a good point. So when you started the study that we recently spoke with Dr. Temporal about, the metacognition and metamemory study with the rats, when you went into that, what were your own personal feelings about what you thought the rats were capable of, or your own personal hypotheses going in?
Going in, I feel like I was a little bit doubtful about if the rats could actually do this, especially when we started collecting data. I was like, I don't know how this is going to turn out. I was hopeful that we would get good results, but I honestly didn't think it was going to come out to what it did. And
I guess my first question on just the rats themselves, just as someone who, I spoke to Professor Jonathan Richardson, Chris, a few months ago, we spoke to him in his office, and we took a deep dive into rats because he's doing all this rat research in South America, doing more in New York City, and it just made my skin crawl, just even talking about the subject on some level, which I know is horrifying to anyone listening to this. But at the same time, for someone who has to, work with them, one-on-one, what was that like? Is that something that you had anticipated a couple of years ago when you were starting your college experience?
Yeah, no, I never thought I would be working with rats on like a day-to-day basis. Not something I was expecting at all, but it was when I first learned about it, it's like, oh, animal research, I thought it'd be cool. I don't know about rats. I've never really seen a rat other than like the ones in the gutter, that kind of thing. But they were friendly. They were cute little pets. Like they're not as gross and icky as people think. Like you're with them every day. You kind of get to know them. They each have different personalities. So it's kind of fun. Yeah.
So that's a great point, the different personality sides. So when you were working with specific rats, did you have like a feel for what they might do or versus what they might not do, or was it fairly uniform throughout?
In terms of the testing?
Yeah.
No, I think that was fairly uniform. It was more like when you're handling them, which ones you had to be careful, they might bite you, and which ones were much more docile, I guess.
Yeah, I would imagine you'd probably want to know the differences.
Yeah, that was very important.
No, that's wonderful. So when you're kind of coming down to the end of the research study and you're preparing the paper and you're potentially being a co-author for something like that, how does that feel as you're kind of nearing the finish line? And then once you publish the paper, what is that experience like? And what feeling did you come away with?
It's very surreal, especially when you look back. And I've spent these long days, long hours in the lab doing this testing. And it's when you're there, it's like, oh my gosh, this is forever. And when are we ever going to get something? And this is going to matter. So it's this, it doesn't feel like it's happening almost. It's like, oh my gosh, how could this be ending? Like I've been doing this for so long and thinking about this and writing about it and talking to Dr. Templar about it. And then it's just very happy and joyful. I'm like, gosh, it's finished and something came of it. And then we start getting like different people talking about it. So it's exciting because it's not really the end of it. It's sort of just the beginning of maybe what else we could do or more research into this field and stuff like that.
Yeah, I can imagine that would be a huge, kind of not only weight off your shoulders, but the sense of accomplishment that comes along with that.
Yeah, I almost can't even believe that it happened. Yeah, I feel very, very grateful to be part of it, and very thankful for all the opportunities I had. But feel very prideful that I was able to help out with this and that it became of something so cool and it got published. And I don't know, it's just I'm very happy. I'm beaming when I got the email from Dr. Templar. It was like the best. I was like, oh my gosh, I can't believe this is fine. I don't know. I was like screaming. I like ran downstairs, had to tell my parents and all my friends. Yeah, it was crazy, but.
Well, you're beaming now, and it's not because you're talking to me. So that's for sure. I can see it right now. And at the same time, that the kind of the time you spent doing this research and everything involved in that is pretty comprehensive. So for students who are considering going that route or exploring their options, there's obviously an opportunity cost along with that. You're going to put all this time in the lab and everything involved with the research and writing it up and meeting with Dr. Templar and all of that. And then you compare it to what else could you be doing with your time. What would you tell for students who are about to make that choice, kind of the positive benefits of going down that road?
I think what you gain from it is just so valuable. Like I learned so much more about how to think critically about different things and how to work through issues and navigate through different problems. And also just my skills at writing improved and my skills at just analyzing data and other sorts of things. The amount of improvement you get from doing that so much more than you would in sort of a normal class is, amazing. And then also, I think it's totally worth it because you get to explore something that you're interested in, that you're passionate about. So it's, even though it is a lot of work, it's fun. It's something that I like talking about. And it makes the day go by quicker because I'm not talking about something that I hate. It's cool stuff. It's fun. It's exciting.
That's great. Well, speaking of class, I know you have to roll. You have a class at 11:30. Yes. We're hitting that time right now. So thank you so much for joining us.
Thank you for having me. It's been great.